everybody, and welcome to tonight's nanoTalks. I'm Carla, and I'm joined by David.
Hi, everyone.
Tonight, we have the pleasure to learn something about two of this year's Nobel Prizes, the Nobel Prize in Economics and the one in Medicine and Physiology. Before we start, I want to quickly give some information about what nanoTalks and also who we, the organizers, are. nanoTalks are short presentations given by young scientists. It's an opportunity for them to present their work, as well as for us to listen, learn, and ask questions. This way, we have the chance to talk about current and relevant topics in an unbiased and objective way. Here you can see some topics we talk about. With this goal in mind, we from REACH organize many different events. REACH is an independent organization that has the goal to bring scientists and the public together. And with this, we hope to spread the scientific facts that may help our society develop. If you're interested to get involved yourself, feel free to join us and get active yourself. There's the link. Okay, now that you know who and what you're listening about or listening to this evening, just some last fact. If any questions come up while listening, feel free to use Slido to ask them. So in the end, the speakers can answer them the best way they can. For this, you can just go to the link in the description box, and you can enter by entering the code nano-nov. Okay, with no further ado, let's dive right into the first presentation.
So thanks, Carla. Before beginning the first presentation, I'm going to give a quick introduction to our first speaker. So our first speaker tonight is Michael Serra-Buriel. Michael is a postdoc researcher at the Institute of Epidemiology, Biostatistics and Prevention of the University of Zurich. And tonight he's going to present us the work that has been rewarded with the Nobel Prize in Economics. And so with all that said, Michael, the audience is yours.
Okay, thank you very much for the very nice introduction. Do you hear me well? Yeah?
Yes, we do hear you.
Okay, perfect. Well, so first of all, thank you for having me here in this nanotalk. And today I'm gonna try to synthesize why they gave the Nobel Prize in Economics to the three winners of this year. And basically, so the winners were David Card, Joshua Angrist, and Guido Imbens. David Card from Berkeley, Joshua Angrist at MIT, and Guido Imbens at Stanford. No? And so what the Nobel Committee said was that 50% of the prize was given to David Card for his contributions to the empirical analysis of labor markets, and the other half was split between Josh Angrist and Guido Imbens for their for the creation of a framework on how to assess causal relationships. So a bit of context before going into some of the examples. 75 years ago, Havelmo, who was a Norwegian economist who also won the Nobel Prize, if I'm not mistaken, said that any theory, any scientist that thinks about the theory sort of thinks it within some testable parameters where he or she can do an experiment. But these experiments are very hard to run in economics, and they were even harder in the 40s. So let's say that in the whole field of economics, there was a lot of advancement and work in theoretical models, but not so much on the side of empirics. And the Nobel Prize of this year and of last year as well has been awarded to those researchers that started to change this trend and what has been termed as the credibility revolution. And basically, it's based on applying the scientific method to economic questions, not only by running experiments. That's why the Nobel Prize was given last year to Esther Duflo, Michael Kremen, and Emil Panel. But the one for this year, more explicitly, was done for how to analyze these economic relationships in a causal way when explicit experiments cannot be performed. And this credibility revolution that is now on full swing in applied economics has put the focus on trying to disentangle causation from correlation from observational data. And now if you go to any economics department anywhere in the world, in any seminar or research presentation of applied work, the focus is on identification of causation instead of purely correlational studies. So now I'm going to show you a couple of examples of the landmark works that started this revolution. The first one, and that David Card and the deceased Alan Kruger, who probably would have won the prize if he had been alive, basically wanted to test a theoretical model about labor markets. So the established theory at the point in time, this was in the 90s, was that if you raise the price of the minimum wage, so if you raise the labor costs, that is going to impact the employment rate by reducing it. So if you make labor more expensive, there's going to be less labor or less employment. And so what they did on this super famous paper was that they compared New Jersey and Eastern Pennsylvania, that were really close to the border of the states. And while in Pennsylvania, the minimum wage did not change over the study period, in New Jersey, on the 1st of April of 1992, it raised by a significant amount, let's say almost 25%, from $4.22 to $5. And the theory that given this price increase or this cost increase predicted that employment was going to go down from New Jersey relative to Eastern Pennsylvania. But in fact, as you can see in the graph, that did not happen. Why this paper is so important is because they tried to mimic the conditions of an ideal randomized experiment where none could be taken place. So basically, they had a treatment and control group, and they evaluated them before and after. And this is called a difference in differences analysis. On another paper that is also a landmark of applied economics, what they did was that in this case was Kruger and Angrist. They assessed how much an extra year of education affected future earnings from work. And because you cannot compare people that have 20 years of education with people that has 10, because they are very different. These students may come from poorer families, or if they have more education from wealthier families, have more connections, etc. What they did was that they used the minimum schooling year laws to compare people that was born and in the early of the year versus late in the year. And because the people that was born later on the year could not drop out of school because they did not reach the age, they were able to mimic this sort of experiment where characteristics of the people that are born late versus early are not that much different, but they attained almost more education if they were born later on the year. And with that, they could see that they had a big impact on the weekly earnings, as you can see in this graph. And finally, so these two experiments would be more on the labor markets and education side of the Nobel Prize. And to sort of finish it, the main reason for which Guido Imbens and Josh Angrist received the Nobel Prize was because they aided in creating a framework to interpret results from randomized trials. So we want to, let's say that we want to look the effect of charter schools on math scores. So if you know, charter schools is a big topic in the US. And because you cannot compare the students that go to a charter school versus a public school or a purely private school, because they are very different, they come from very different backgrounds and so on. What is done is that because there are some students that can access to the charter school through a lottery process, that is a big deal in the United States, you have a sort of a randomized experiment in which by winning the lottery, you are able to access a charter school, and you have a control group that is equal in terms of their characteristics to the one that won the lottery because it was randomly assigned, but did not have the chance to attend school. But there are three things here. So we have the effect of winning the lottery on actually attending the school. We have the effect of attending the school, the charter school on scores, and then we have the effect of winning the lottery on scores. The first, so winning on attendance, is observable. The last is also observable, but the one that is not observable is actually the effect of going into the school. So by simple substitution, by dividing the effect of winning the lottery on scores by the effect of winning on attendance, one can sort of approximate this effect. So for instance, here we have the distribution of scores of the students, and we see that so 371 students applied to this lottery, 253 won and 118 lost. And because it was a lottery, they are completely equal in terms of their characteristics. But we see that that already has an effect on their scores, on their math scores, more explicitly by 0.36 standard deviation. Then the other thing that we see is that not all those that won the lottery actually were enrolled in the charter school, and not all of those that lost the lottery did not go to charter school, but we see that the probability of winning the lottery on actually going to the school increases by 74%. So using the framework from before, we can estimate the effect of attending on scores for those that actually won the lottery and went, and those that didn't win the lottery and didn't go. And you can see that it's bigger than the effect of winning on scores. It's almost half a standard deviation. And so the math and the statistics behind this framework comes from an econometrica paper from 1994, which is called the Identification Estimation of Local Average Treatment Effects. And basically what it says is that, this is called an instrumental variables approach, that if you want to look at the effect of charter school attendance on math scores, you cannot assess it, let's say, just looking at the data, because people that go to charter school are very different from those that do not. and that biases the relationship. So what you need is that you need an instrumental variable, which in this case is the lottery. And with this, you are able to remove this compounding effect. So this first step is called the validity of the instrument. So the first condition that you need for the calculation that I showed you before to hold is that winning the lottery has to predict whether you go to a charter school or not. Then you also need the lottery to be completely uncorrelated with these factors. And then you need that the winning the lottery only affects math scores through charter school attendance. So let's say that if students that won the lottery were suddenly more happy and super motivated to study more, and through that they got great scores, the calculation that I did before would be biased. This is called exclusion restriction. And this has huge implications, not only for economics, but for all the rest of applied fields, because it is what is used to adjust in clinical randomized trials for non-compliance with the randomized treatment. And with this, I'm done, and I'm now happy after the other talk to answer your questions. Thank you very much.
Thank you very much, Michael, for this very interesting presentation. I'm sure we'll be able to go into some more details later during the Q&A session. And by the way, quick reminder that in case you have a question, please post it to Slido, and we'll try to ask it to the speakers during the Q&A session at the end of both talks. And so with that, Carla, I'm giving you the mic back so you can introduce our next speaker.
Yes, Noemi Fresel will give us the second presentation about the Nobel Prize in Medicine and Physiology. Noemi is also a postdoc, and she's also a member of REACH. She therefore has much experience at the work we are doing, so to say, moderating. It's her first nanoTalk today, though. So I think there's no more to say. The stage, or maybe rather the screen, is yours, Noemi.
Thank you, Carla, for the introduction. And let's see how it is to be on the other side of the screen. So I will share my screen. Okay, sorry. I think I did it too fast. I'm sorry, my presentation is not in the sharing option anymore. All right, this is back. Okay, sorry for the delay. So I will try today to give you an idea, explain you a little bit why these two researchers, David Julius and Artem Papaputian, got the Nobel Prize in Physiology this year. And so first, the short answer from the Nobel Committee is because their discovery explained the molecular basis for sensing heat, cold, and mechanical force. So David Julius found the receptor for temperature and heat pain, and Artem Papaputian and his team found the receptor for sensing mechanical force. So what does this mean, though, molecular basis? That means that they actually found the molecules that are expressed in our sensory neurons and other cells that are able to detect the stimulus, such as temperature, pain, touch, mechanical force. So finding these molecules alone so was already quite a feat, but why are they important? Why are these the molecules so important? Of course, to perform everyday activities. And if you might not like this one, you probably do this one a lot every day, or at least at the beginning of the day. If you take the first few minutes, maybe you get up, grab a cup of coffee, bring into your mouth and try to drink. And usually this happens without a problem because your sense of touch and proprioceptions are working properly. So proprioception is basically the ability that you have to sense where your body is in space. So for example, in this instance, it will be know where your arm, your hand, and your mouth are. And this allows you to do the task properly. And you can try this now at home, even though it's a bit too late for coffee. You can close your eyes, bring your finger to your nose, and then to your elbow, and then to your nose again. And this is quite easy. And you can do this because you have receptors in your joints and your muscles that are able to sense the load, the stretch of the different parts. And all of this information is sent to your brain that can then tell you where your arm is, also where your nose is, and then you're able to do this task. And the way that all these receptors detect load, stretch, all these mechanical force related things is because of this molecule piezo2. And this is also very important for sensing touch. So where you grab the cup basically is also important. So maybe now you don't do this so well, drink too fast, spill a little bit, you might feel the heat, maybe a little painful even. And so this is possible because some other sensory neurons have this other molecule that's called TRPV1 that is able to sense the heat and especially the painful heat. So now you might already get a sense of how important these molecules are for explaining our everyday function of our bodies and activities. So already like discovering these molecules is a good enough maybe to get a Nobel Prize. But if David Julius and Artem Papapeutian got this Nobel Prize, it's also because the methodology that they used and and the experiment that they did were very smart and innovative, and they allowed a lot of researchers afterwards to find similar molecules and also to describe the neurons that have these receptors, how they communicate to each other and to the brain, to basically explain how we sense all these stimuli such as heat, cold, pain, pressure, and so on. So let's take, for example, the case of this heat sensitive neuron. How does it work and how did they find this TRPV1 molecule? So in the 90s already, some researchers found that sensory neurons that are sensitive to heat, when they feel the heat, they produce an ionic current. And this is basically electrical activity. And this is a way for neuron to transmit information. And this is how it's going to be able to send it to spinal cord and brain. And then you will have this perception of the heat. But we don't know how the heat is detected by the neuron. We also know that there's this chemical compound called capsaicin that is in chili peppers. And this also detected by the same neuron and also felt as hot. So maybe the idea is that the heat and this capsaicin molecule, they activate the same receptor. And this is how the neuron thinks that chili pepper is hot. And so if it has a receptor that can detect these different stimulation, that means there's a gene that encodes for the receptor. So the idea is we're going to look for this gene. And what they did is just they took many genes of these heat sensitive neurons and put them not exactly one by one, but almost in cells that otherwise would not be sensitive to heat until they tried this with many genes until they found one that was called TRPV1. And if now the cells would express this gene, so would have this receptor, they would be able to open and generate this ionic current when you put the heat more than 43 degrees, and that's painful heat. And they would also produce this ionic current if you put on capsaicin on them. So this is how they identify this TRPV1 as the receptor for heat, and in particular painful heat, so more than 43 degrees. And then by analogy and similar experiment, they were also able to identify it in the next years a lot of other receptors from the same family that basically responds to various ranges of temperature, and also explain how we sense more extreme or more mild heat temperatures. And they also find out, for example, that menthol activates the receptor for cold. So this is also in the same way that capsaicin feels hot because it activates the heat receptor, menthol feels cold because it activates the cold receptor. And then in the next years and decades, this TRPV1 was also showed to be important for a lot of other heat and pain related phenomena, such as regulation of body temperature, also inflammatory pain, neuropathic pain, visceral pain, so that's pain from the gut, and also in protective reflexes. And like I said, they also use the same methods to identify a lot of other receptors from the TRP family and also from other families of receptor that explain how our neurons can sense various kinds of stimulation and also various kind of chemical compounds. But one of them was still missing. And if we come back to the example I was talking at the beginning about, you're holding the cup, you're feeling the texture of the cup, you know where your arm is, you can drink, maybe walk at the same time without looking at the cup, without tripping on the floor. And for all of this, you need, or your organs and your cells need to feel mechanical force. You need to feel the touch of the cup. You need to know where your legs are, if you're walking properly, and all of this requires that your cells have a mechanical sensor molecule. And this is not trivial, right? People who don't have this, they're really unable to perform this super easy task that you tried maybe before. And for us, we don't really think about them, but these people who have deficiency in this molecule, for them, it's really, really complicated. So everybody knew that this molecule is super important, but it was very, very hard to identify. And other reasons why we're really, really anxious to find it is because you have a lot of other cells or systems in the body that might require a pressure or mechanical force sensor. And that might be, for example, your red blood cells to circulate properly in the vessel, also the vessels to change diameter and regulate blood pressure, your bones, for example, to sense mechanical force and stay solid, synthesize some more bone whenever it's necessary or not. Also, your bladder needs to feel the stretch, and that's also mechanical force to tell you to go, but don't go right now. So we really wanted to find this molecule. So what Arden Patapaputian and his team did is, so they thought, like previously for the heat sensitive neurons, I told you if it's able to feel the heat, it has a gene that encodes for a receptor to feel the heat. So now they reason the same. We want to find out which gene codes for the receptor for the mechanical force, because finding this receptor alone is very hard. So first they found some cells that they had in culture that were able to respond to the mechanical force. So, and by respond, we mean that they were able to generate this ionic current, so we can measure electrical activity. And once they had these cells, they said, okay, so there is the receptors probably are in the cell. We're just going to silence one by one, and this time they really did one by one, genes that could be the code for this mechanical sensor protein. And they silenced them. And after 71 trials, so maybe a year and a half, 2 years, they finally found a gene that when it was silenced, the cell would not respond anymore. So that means the mechanical force receptor is not there. And they named this gene piezo1 because piezo means pressure in Greek. And then by looking at other cell types and other organs, they also identified piezo2 that is quite similar and is found out then to be very important for sensing the sense of touch and proprioception, for example, in the mammalian body, so humans and other mammals. So this is then after finding the receptors for heat and pain, and then, like I said, many other ones for a lot of chemical compounds, we finally had, and this was in 2014, so not that long ago, we finally had the receptor that is able to sense mechanical force. As I said, it's important not only for touch and proprioception, those are the main function of this piezo2, but also it's been shown to be important for mechanical pain, urination, respiration, blood pressure, skeletal remodeling, so that's bone remodeling. And this list is still growing larger and larger every day because this research is pretty recent. So that's why, in short, these guys, David Julius and Artem Papapoutyan, got the Nobel Prize, and they shared it because their work describe two very important functions that explain how our body is able to feel, interpret, and interact with our environment. And if I may keep you for another second, I think it also shows how much fundamental research is important to the understanding of our physiology, because David Julius just was curious to know why chili peppers feels hot. And then about 20, 25 years later, we now understand how our neurons are able to feel our environment for a lot of wide range of modalities, and we understand how our nervous system works, basically. So I think this is a good reason to get the Nobel Prize, and I hope you have a better idea now why they got it and why I think it was a good idea. And thank you very much for listening, and thank you to the nanoTalk team to have organized this evening.
Yeah, I think we definitely have a better idea on why it matters. So thank you very much also, Noemi, for this very interesting talk. So maybe now is the moment, the opportunity to ask questions to the speakers. So maybe I will begin by asking this first question, maybe to you, Mikel. So it's a bit of a broad question, but some of those works and studies you talked about have been done more than 20 years ago. And so what would be interesting to know is, nowadays, are there consequences or example of improvement in like public policies that can be arguably linked to that work, in your opinion?
Yes, but most importantly, the contribution of these people is that basically all papers that are written nowadays in applied economics use these methods. So there are no purely correlational or more, like uncontrolled studies. And that's, to me, is the biggest contribution. Like, they really have changed how the whole discipline works, all of them. And in terms of practical implications, yes, like you can now, one that I think that was published, I think last year by some economists at ETH, was that when Switzerland opened its borders for foreign high-skilled workers, actually the wages of the Swiss natives increased due to that. So it's not only about evaluate, so sort of saying, okay, what we have to do, but it also serves as an evaluation tool of things that have happened in the past, and within a causal framework, which is what we really want in science at the end. So I think that's the major contribution to changing the whole field.
Okay, pretty impactful. So yeah, all right.
Okay, then we go to Noemi. One question. I think it's just, or it's already important to find out, or yeah, which genes are in charge of proprioception and temperature. But I think in medicine, the main thing is also like not only to find out how things work and as a consequence how things don't work, but also to treat them. So is there already, based on this discovery, some treatments that are used today, or is there nothing like this yet?
There are. I'm not going to go a lot into specifics because I'm not super familiar actually with them. There's a lot of leads for various chronic pain conditions, but the translation is very hard. I think for treating heat pain, it's a little bit better. One of the problems, for example, now that, so all of the studies with piezo2 are very, very recent, because it's basically in the last 5 years that they've been going on. One of the main problem with this is, as I showed you, these molecules are expressed or present not everywhere, but really in a lot of different cells. And so you cannot really make a treatment that would target everything, because you would have a lot of side effects. And that's a lot of the problem with the pain treatments. And so the goal and what these researchers are aiming for is to find maybe local treatments. And that depends if you have what they call gain of function or loss of function. So if you have too much or too little of these molecules. And then the idea is to try to find like local ways to change how they function. It's a bit long maybe, but basically the thing is also, if you identify all of these molecules and all of the different compounds that it can respond to. So for example, TRPV1 responds to heat, but also to capsaicin, and then maybe you can use capsaicin to activate those neurons if you want, right? So you can. So that's how understanding the basic physiology of it can help produce treatments. But this is a very, very active and ongoing field of research. And it's really, really difficult to translate from research to medicine, basically. But it works sometimes.
Thank you.
So we have questions on Slido. So maybe I will ask you that, Michael. So regarding minimal wage, is there a general consensus on how much it would influence employment, like in general? And also the second part of the question would be, why, in your opinion, does Switzerland not have a minimum wage? Like I would imagine because of those studies, maybe it would make sense that we would have one so far.
Okay, so it's actually not my area of expertise at all. So I'm gonna give here pure speculation based on my readings here. I mean, I think the consensus is that minimum wage has very small effects on employment, if any. There are some meta-analyses on the topics. Where you can compare different contexts and so on. So it really doesn't seem to have a lot, to reduce employment a lot. And regarding the minimum wages in Switzerland, I think that Switzerland is kind of characterized by a really strong belief in the free market, given its institutional context. And I don't like. The unemployment is so low that it might not even, make sense to have this. And also, like, if you compare it in real terms with any other country in Europe, it's, both in terms of real wages and in terms of unemployment, it's ridiculous. So yeah, that's my take. But again, I'm no expert, so don't trust me on it.
Thank you.
Okay, there's another question to you, Noemi. The question is, how do you see if a cell feels heat Like how is it reacting? And also, how do you find cells that don't react to heat? Like, how do you notice that?
Yeah. All right. So there are basically two ways to, when we say a cell detects or reacts, there are two ways to measure that. So I'm talking mainly about neurons, but this is sometimes also true for other types of cells. One of them is that they will produce an electrical current. And this is especially the case for these two molecules that I described because they're channels. So basically when the channel is closed, and then when you have the stimulation, can be heat or mechanical force or piezo, the channel opens and some ions that are outside the cells get in the cells. And this creates an electrical current, basically. So you can measure this with putting like an electrode into the cells or near the cell. And another way to say that the cell responded or reacted is by looking at calcium ion fluxes, because also when this channel will open, you will have, so calcium ions can get in, like I said, and you can also have other reactions like chain reaction that will lead to an increase in calcium inside of the cell. And this we have sensors that can sense when there is more calcium. And this is called calcium imaging. And it's a proxy of the activity of the cell. So you look at the calcium level, and then when the calcium goes up, you're like, oh, the cell reacted. And so depending on what receptors the cells have, they might respond or not to different types of stimulation. So the cells that have TRPV1, they will respond to high heat or capsaicin, for example. The cells that have TRPM8 will respond to cold, but if they don't have TRPV1, they will not respond to heat. So it's very specific to which receptor they have.
Okay, thank you.
Maybe another question that I could ask to you, Mikkel, would be, so in the descriptive text of the Nobel Prize that we can find on the Nobel Prize website, they often mention natural experiment versus regular clinical trials. But so could you maybe give us your insight on what's exactly the difference between those two and what's so revolutionary about what they did in the field of that Nobel Prize?
Okay, so a regular experiment is an experiment in which the researcher explicitly modifies and randomizes the cause or the treatment and observes an effect or the absence of it, okay? But explicitly randomizes this. So for instance, let's say that we were going to do an experiment on pain, okay? So how much pain do you feel when you get stabbed in the leg? If you wanted to do an experiment about that, what you would do is that you would, randomize, have a group of 100 volunteers, and randomly stab half of them and compare the difference between the ones that were stabbed and the ones that weren't. Okay. What happens? This clinical trial and many other more are not ethical to do or to even propose. So what happens is that there are some cases in nature where there is this sort of randomization, okay? That happens outside the control of the research, but it mimics the conditions of a let's say, a standard experiment. So there are many examples, but for instance, one that is pretty cool is, okay, how does pollution affect hospitalizations? Okay, so ambient air pollution. Because people that live in more polluted areas tend to be poorer, they have lower education, and so forth. You cannot simply compare, people that have that live in high polluted areas versus people that live in low polluted. So what these researchers did, I think one of them is Tatiana Derivina at the University of Michigan, was that they used an instrument, a variable that mimicked the random assignment. And this variable was the direction of the wind. Okay, so how the wind blew northeast, northwest, or in whichever direction in all the cities in the United States. So they did not compare the people that lived in high versus low polluted areas, but they compared people that were exposed to pollution only because the wind was blowing in a certain direction on a given day. And that mimics an experiment, but the researcher cannot control this random assignment. And the other thing, so the contribution that Guido and Josh were also awarded for is that you can have natural experiments, so that the LATE framework, the IB instrumental variable framework can work both for analyzing these natural experiments that I just talked about, but also can be used to analyze randomized clinical trials with imperfect compliance of the treatment. So let's say that you randomize half of the people to the studying experiment, but only half of the people that were assigned to the start condition actually get start. So that's why their methodological contribution try to adjust for that.
Okay, thank you.
Okay, another question to Noemi. There's somebody who wants to know what other aspects of environment the cells can recognize, and also maybe, I don't know if you know that, but if there are some researches about animal and plant cells, and if they work the same way, as they are, like, especially plant cells are quite different from human cells, if there's some research about things like this.
Yeah, so I'm going to maybe deflect a little bit the question about the plants, because I'm really not familiar. They have also sensors for chemicals and mechanical force, for sure. They're not really the same molecules. But then when you go to animals, and especially all of the molecules that I was talking about before, they've been identified in mammalian cells, and these will be, really, if you take like a mouse or something, they will be very, very close to what you find in humans. So that's why a lot of this research is done in mice, because you can also activate, inactivate pretty easily either the receptors or the cell to identify what does what, and then compare to how it is in humans. So these will be quite close. The molecules themselves, the gene, and also the effect they have. But I think they will be as a whole quite different from the one and the mechanism that exists in plants. The other part of the question was other parts of the environment that we can sense. Well, a lot. So that depends on the sensory system, right? Sensory system will sense what's outside of your body, so your environment. And so everything that I talked about is part of what we call the somatosensory system. And you have other sensory systems are like the visual system, for example, or the hearing system. So sounds and images are stimuli from the environment. The somatosensory system will sense everything from and around your body, basically. So for example, if you stay on the somatosensory system, I've talked about heat, so cold and general temperatures, some chemicals, and chemicals are super wide variety. And then there was this whole mechanical force aspect. In chemicals, for example, what might talk to you is everything that itches, for example, or also different molecules or chemical compounds that will basically arrive on your skin, and then they will activate receptors like the one that we talked about before that are specific for this kind of molecule. For example, histamine is a very potent itchy molecule, and you have some receptor that responds to histamine, and this will then be very itchy. And yeah, what else? You have lots of different molecules. For example, ATP also, if it's injected in the skin, will be super painful because it activates also pain neurons. Yeah, basically everything that you can think of that you feel from your environment, you have a neuron that is adapted to sense it somehow.
Maybe then a little bit further, if there's something that is you cannot feel, like for example, UV radiation, does the cell have receptors for that and we just can't feel it, or does it just get destroyed?
It's not destroyed. I don't think there's like a receptor for UV radiation, but that doesn't mean there's not an effect on your cell from UV radiation, right? For example, it will go through your cell and up all the way to the DNA that's inside the nucleus and damage it, right? So it's not doing nothing, but it's not sensed as a stimulus for, oh, this is happening outside of me, I'm feeling it, now we'll act, right? And yeah, but you have, I mean, UV is just another wavelength, right? You have receptors for some wavelengths of light, and those are the ones that we can see, that they're in your eyes. And you have some wavelengths like outside of the spectrum, UV and infrared, for example, we cannot see them because we don't have the receptors for this, but other species may have it. Right? So it depends. You can basically sense what you have receptors for.
Okay, thank you.
Okay, maybe a last question I would have, that is on Slido. So at the beginning of your talk, you wrote a Nobel for the credibility revolution in economics. So could you just give some more details about what you mean by credibility revolution? That's the question.
Yeah, so the credibility revolution in economics has been basically the shift from theoretical results to causal effects of interventions. And it's called credible because before this happened, the most applied papers were using purely correlational methods, not natural experiments or this sort of research designs. And what has happened now is that this has become the norm. If you want to publish a paper, an applied paper in an economics journal, you will have to have thought very, very carefully why your correlation is causal. If it's like if there are any holes in it, you will not get it published. So it's become the standard of the whole field, and it's called the credibility revolution because it stresses how important it is to have credible results that are not confounded by unmeasured factors.
Okay.
Okay,
Nice.
then to be fair, a last question to Noemi. Do you have to silence one gene? Do you have to go through the whole DNA, or are there and block every single gene, or is it are there some areas that are more likely to have the gene that produces the protein they were looking for, or do you really have to go through the whole DNA?
So basically the whole DNA in every cell, independent of the type of cell, in everything is the whole DNA for all the genes of the body. Some of them are expressed and not. And so what they did in Ariam Papapoutyan's lab, and it's quite common to do to silence some genes like this, is that you will target the gene that you want to silence. So you know the sequence of the whole DNA, you know where are the genes. So basically you have the sequence of letter that is the gene that you want to aim for to silence. And so you will build what they are called RNA interference or interfering RNA. And this is, as you might have heard a lot about message RNA this year or the past year and a half. This is another kind of RNA, but the principle is also the same. It's a sequence of letters that will be able to go and target the part of the DNA that you want. And it will interfere with expression. That means it will prevent the protein to be synthesized from the genes. And that will, so the DNA is not changed or anything. Like it's just that the protein is not going to be expressed at the end anymore.
But how then do you know which genes to silence? Like, do we know as human beings every gene and its function? Probably not.
So we don't, but more and more and more, and that's also probably, it's a lot of technical advances, but why it took only 72 genes to silence in 2014, when in 1997 they had a pool of 60,000 genes to try out, I think, because now we know, basically we have the whole sequence of the genome, and the more and more we know some of the genes, what they code for, the more and more we're able to guess or to predict from the sequence what others that we don't know what they do, what they might do, right? Because a lot of genes, sometimes they code for proteins that kind of look alike, or they're the same family. For example, with this TRPV1, there's a lot of other TRPV with other numbers, and other TRPM and TRPA, and they have similitudes between these proteins. That means the gene also look the same. So we say like, oh, this one is also a TRP something, so it might also do a function that is related or similar. And so I guess what they did in Arlem Papapoutian's lab is they didn't choose 70 genes out of 21,000 just by chance. They had a wish list or a priority list of like, these genes, they're good candidates because we know that they might make a protein that looks like something that's what we're looking for, because we know the sequence of the whole genome and we're making guesses based on what we already know from other genes.
Okay, perfect. Thank you.
Okay, so I think we can end the Q&A session here. And with that, that's going to be it for tonight. So again, we'd like to thank you, Michael and Noemi, for taking time tonight to inform us about those Nobel Prizes. Thank you very much. I'm sure we all learned a lot, and it's always very interesting to see each year the different Nobel Prize and what are the consequences of those works. And yeah, very useful to know more about the state of the art in those subjects. And thank you very much, everyone, for attending this Nano Talk tonight. Thank you, Carla, for moderating that together. Before you leave, please let me remind you to subscribe to this YouTube channel if you haven't yet. And please note that there is going to be a new Nano Talk event on January 27th. So stay tuned. And yeah, I hope you'll join us again for future Nano Talks. Thank you and have a nice evening. Bye, everyone.
Nice evening. Thank you. Bye.
Thank you.